I just want to kind of highlight, there were a couple questions kind of like the formation of life on Earth. So I want to talk about that. I want to talk just briefly about viruses. We've kind of mentioned viruses when we talked about DNA and bacteriophage. We'll talk a little bit more about viruses. And then also just kind of talk a little bit about bacteria. You will not be quizzed on this material. This is just kind of like, hey, just in case it's on the EOC exam, like we'll kind of get to it. Earth's early atmosphere. These are the gases that were present in Earth's early atmosphere. First of all, how old is the Earth? 4.5 billion years old, somewhere around there. How old is the universe?
11 billion.
Around like 15 billion years old. 15 billion years old. All right. So, anyways, here's Earth's early atmosphere. What is it missing? Yeah, it's missing free oxygen. Now, there's oxygen, carbon dioxide, H2O. Like there's oxygen here, but it lacked oxygen. The question, and this is a very kind of fuzzy part of biology because we don't know what the answer is. We don't have the right answer for how life formed on Earth. There are people who say that it came from a meteorite. A meteorite struck with microbes, and that was the start. That's great. But then how did the life start on the meteorite, right? So that is a possibility. Some people do believe that. I don't know which way it is. I'm kind of more of the feeling that life started here on Earth and it did start on a meteorite. But in any case, we're trying to answer the question, like, how did life begin? Where was the first, like, true origins of life formed? So in any case, here are the gases, right? Hydrogen, carbon dioxide, methane, N2, SH2, sulfide, H2O. Really, these molecules, these small little molecules, are really all the components we need to make all of our macromolecules, right? So the starting of life on Earth has to start with the formation of macromolecules. In order to have life, we need to somehow figure out a way how proteins were made, or lipids were made, or carbohydrates were made, right? All of these different things. Nucleotides were made. So this is the starting point. Miller and Urey, it really started off this guy named AI Operating, who's not up there. But AI Operating really proposed this kind of primordial soup concept that Earth's early atmosphere wasn't a nice place. We wouldn't do well there. It was really, really hot. There were a lot of volcanic activity. There was a lot of like worldwide storms and lightning. It was a volatile place. So AI Operating proposed that the energy in Earth's atmosphere, like the lightning and the heating and the evaporation and the cooling, could have possibly formed these molecules into our first organic molecules, right? Our first amino acids. So he really proposed it. It was Miller and Urey that really kind of devised an experiment to see if this was even possible. So this is their experiment right here. Here's the water represented here as oceans. The gas in this chamber are the gases represented by Earth's early atmosphere. So carbon dioxide, methane, sulfide, right? Everything on that previous slide. Well, the sparks represent the lightning energy in the atmosphere. The lightning energy in the atmosphere. And then the heat just kind of represented the heating process that it was formed. It's hot. Like the oceans were evaporating. And in this experiment, they kind of ran this experiment for a period of time, and they had this collecting trap. So they cooled it down, condensed it, and they collected some materials in here. And over weeks, months, they actually found some very, very simplistic amino acids. So what's interesting about this is they showed that we really have the possibility of forming amino acids. Yeah. It blew up. But the town is usually called it. I'm like, I don't know. All right, so here we go. They formed amino acids. I know that sounds like, hey, so what? Think about that. They formed amino acids. They formed the components of proteins. Later on, they showed that they could form RNA molecules. If this shows the possibility of forming these organic molecules, it is a huge step forward for the possibility of how life formed on Earth. We don't know exactly how we got from here to little bacterial cells, but at least it's a step. It's a step that was shown scientifically with data that actually this could happen. Okay? That was the first step. Next step, you take oil. All right. I'll show you real quick, right? You put oil in water. Right? Oil is a lipid. What does it do? Does it dissolve?
No.
No. It does not dissolve. It's hydrophobic. It dissolves up. So I take a little bit of oil, put it in water, and you can see what it forms. You can come kind of take a look real quick. But it forms kind of all these little balls. See it? All right? If you want to come by and take a look, it actually forms one large one. It forms a lipid circle. Right? Why? Because the lipids are trying to get away from the water. In essence, it kind of just formed a membrane. It did. It does it naturally. So the concept is there were lipids that were formed by this process, and lipids naturally form membranes. This is not a living cell, but it formed a membrane, and let's say it happened to form some kind of simplistic enzyme in there that starts just kind of breaking down some sugars. Right? We start having some metabolic activity in this. Is it living yet? No, but we have it just metabolic activity is just breaking down chemicals. It just has some chemicals being broken down within the lipid. What we need to do is we need to get some DNA component. I said this before. We don't really think that DNA was the first molecule inheritance. We think RNA was the first molecule inheritance. Why? DNA can hold genetic information. RNA can hold genetic information in what? Lysosome. It's acting like a what? The big E word. It acted like an enzyme. So RNA has some enzymatic qualities and it can hold genetic information. That's a pretty big deal if you're a really simplistic life form. If I just get an RNA molecule in here, you now start going to see how life came together. I know it's vague. I'm not really giving you specific details. We don't know what the details are. We're kind of like proposing this is the process. So eventually, life forms. The first life form. Do you think it was anaerobic? Had to be anaerobic. Why? There's no oxygen. So the first life form, it was a heterotroph. I know if you're saying, well, if there's no oxygen, it had to be photosynthetic because what's it going to eat? It wasn't it wasn't an autotrophic organism. The first organism was a heterotroph. Okay? And it was an anaerobic heterotroph undergoing glycolysis. That makes a lot of sense because every heterotroph in the world undergoes glycolysis, right? It's in everything. So it kind of makes sense to this. So it's an anaerobic heterotroph. Now, they're a heterotroph. What are they eating? If there's nothing else to eat, well, they're eating organic compounds. They're eating sugars, protein, but that's also eating as well. I know it kind of seems a little different, but it's actually eating it too. Now, these anaerobic heterotrophs were producing a whole bunch of CO2. There's a lot of light available. Nothing's using the light, which means there's this huge void, this big niche that an organism could take advantage of. And eventually it happens. And our next organism was an autotroph. A photosynthetic bacteria, also called like sometimes cyanobacteria. A photosynthetic bacteria now undergoing photosynthesis. They had a field day. Was there anything slowing their growth? No, there's tons of nutrients. There's nothing slowing their growth. There's nothing eating it. So what did they do? They produced a whole bunch of oxygen. There's a whole bunch of oxygen. Hey, guess what? Do anaerobic bacteria typically like oxygen? No, they don't. They typically don't like oxygen. So we started to produce an atmosphere that the anaerobic bacteria didn't necessarily like. Some can survive, some are back to those anaerobic can survive. So. But it also created a whole new niche to be occupied. What's the next organism to come about? An aerobic bacteria. So now which is the ancestor of our, right? Not all of them are, but one of them was the ancestor of our mitochondria, right? Because now they're far more efficient. Remember, aerobic respiration produces about 36 ATPs per glucose compared to just 2 ATPs. So now they occupy the niche. Life is off and running because now we have this very efficient energy pathway that we can use, aerobic. All right. Now, all of our organisms right now, they're all simplistic, they're all unicellular, they're all prokaryotes. What's the next step in the evolutionary chain to form a eukaryote? And I think I mentioned endosymbiotic theory before, have I? In class? The big cell eats a little cell. So endosymbiotic theory happened twice. Right? We know this is true. We have evidence of this because mitochondria have their own DNA. Mitochondria actually replicate on their own inside your cells. So at one point, our ancestral mitochondria were their own organism. Right? A larger cell would engulf them. When they engulfed them, they didn't break them down. They actually used them to make energy for them. Eventually, at some point, the mitochondria inside and the cell itself started dividing together. They would divide, and it's now kind of part of the single life form. Eventually, did we have to form a nucleus? Yeah, a nucleus formed, the DNA was organized a little bit more. Other organelles were formed like that, all part of this as well. But really, the key starting point of endosymbiotic theory is the taking in of the mitochondria. Endosymbiotic theory happened twice because it also happened with chloroplasts. So, this endosymbiotic theory happened when we got our first eukaryotic heterotrophs, and it happened again when we got our first eukaryotic autotrophs. Everyone okay? You want me to stop there and we'll talk a little bit on Monday, or you want me to just power through? People want to practice? Let me just say this real quick. I'll go real quick. Viruses, are they living?
No.
No, they're not living. Or for viruses to reproduce, they must take over a host cell. We already know that. Here's a bunch of viruses right here. You can be different shapes. So here's a mosaic virus. Here's an adenovirus, influenza virus. Here's a bacteriophage. We know the bacteriophage well. Right, so just some different viruses. There's not a ton to know here. What I would know is just kind of this. There's two main life cycles of viruses. You do not need specifics on this. I don't think this would be on the UC exam. I'm just mentioning it in case you see the terms, you have an idea what it is. Viruses can either send your genetic information into a cell and immediately start making new viruses that cause the cell to lyse and explode. Or viruses can actually integrate their DNA into the bacterial DNA. A large percentage of our DNA is actual viral DNA right now from all of the viruses that have infected our ancestors through the thousands of years of human existence. Even what has to be existence, one of our precursors. Right? So this is known as the lytic cycle, when the virus is actively making more viruses. This is just known as the lysogenic cycle, when the virus genome is just incorporated into the bacterial and the host genome, and when the host cell replicates, the virus genome is replicated as well. They can shift between the two. This, with certain factors, can shift the lytic cycle, 2 years, 3 years, 5 years down the road. Hence, you can have a virus that's sitting dormant. I think it's kind of key with, I think that herpes virus does that. I believe, because it kind of comes back every once in a while. All right, that's it. One last thing about viruses. Not all viruses are DNA based. Some viruses are RNA based. We call them retroviruses. So a retrovirus, this is the last thing. A retrovirus is an RNA-based virus. So in order for a retrovirus to attack your host cells, and give an example of retrovirus is HIV. In order for HIV to attack your white blood cells, it needs to take its RNA genome and convert it back into DNA. That's done by an enzyme known as reverse transcriptase. Again, you don't need like specific terms here, but if you see reverse transcriptase, you kind of know what it is. Reverse transcriptase is mistake prone, which makes HIV very hard to treat because it's making mistakes, it's making mutations. Mutations allows HIV to evolve, to change very quickly. So therefore, HIV is hard to really kind of nail down because it's constantly changing. You get new strains of HIV all the time. Because it is a retrovirus and reverse transcriptase tends to make mistakes in the process. So it's just kind of an interesting thing. All right, we're done. There's HIV right there. Kind of an interesting note on HIV. Some people are immune to it. We kind of found this out in a weird way. Not going to go into details right now. But some people have HIV needs to bind to certain receptors on your white blood cells in order to gain entrance into the cell. Some people have a slight mutation in one or two of these receptors. Their white blood cells are still functional, but because of the mutation in the receptor, HIV can't bind to it. So there's a small percentage of our population that's immune to HIV, which we've learned about. It's kind of interesting. Yeah.